Hey everyone, welcome back to Windows Seat Studies. I hope you all are doing well. We were discussing about cancer biology, in which we have covered the two important hallmarks of cancer in detail. So, we discussed about how the tumor cells they are able to sustain a proliferative signal, which is going to lead to the uncontrollable cell division. And we also learned about how the tumor cells they are able to evade growth suppression, which will lead to the cancer. So in the last class we discussed about the interplay between the retinoblastoma protein and E2F transcription factor. So we learnt about this tumor suppressor pathway, which when is dysregulated leads to the cancer. In today's lecture, we are going to discuss about p53. Okay, which is an important regulator of cell cycle progression. So this p53 protein, this is encoded by gene known as TP53. Okay, and this p53 this is regulator of cell cycle progression, genomic stability, this is regulator of genomic stability, and apoptosis. So this regulates all those three processes. The p53, the wild type p53, this regulates cell cycle progression, genomic stability, and apoptosis. The cell cycle progression from G1 phase to S phase is regulated by this P53. So this P53, the wild type P53, upon activation, this causes cell cycle arrest. Okay, and why it is important? Why the wild type one is going to lead to cell cycle arrest, and how this is so this cell cycle arrest it is caused by p53, and that is why we call this as a tumor suppressor gene. Okay, because it is going to lead to the suppression of tumor, so it is going to keep a check that the uncontrollable growth, uncontrollable proliferation does not happen when it is functioning properly. So once this p53 it loses its function, the wild type p53 loses its function, it is going to lead to the cancer due to the uncontrollable growth. So let's learn about p53 in detail. How does it lead to the cell cycle arrest and what does it do? So normally in the cell which has normal genome, which is functioning normally, the p53 it is inactive. It is in the inactive state. It is actually degraded. So there's a protein known as MDM2. Okay, so this is p53. So this MDM2 is it ubiquitinates p53. Okay, so due to the ubiquitination of p53, p53 will be degraded. So this will be degraded. In the normal condition, when cell is normal, it is functioning very normally. Okay, but whenever there is some DNA damage, or let's say there's some stress such as oxidative stress, or cell is undergoing any kind of stress, so there is some cellular stress due to radiation, or let's say the oxidative stress, in all of those conditions, that is going to lead to the double stranded breaks in the DNA. Double stranded breaks are they're very fatal to the cell, because that is going to lead to genomic instability. So this double stranded breaks, these are sensed by the kinases known as ATR ATM kinases. So those are the kinases which are going to sense that the DNA has a break. Okay, so the double stranded DNA breaks are sensed by ATR ATM kinases, and now they downregulate it, downregulate a kinase known as CHK2, and this kinase is going to activate MDM2. So sorry, this kinase is going to phosphorylate the P53. This phosphorylates P53. So due to phosphorylation of P53, now MDM protein cannot, let's not draw this inhibition arrow. Okay, so MDM 2, this cannot work. This cannot recognize the p53. This can only recognize the hypophosphorylated p53. So once the phosphate group is added on p53, MDM cannot ubiquitinate it, and there will be no degradation of p53. So we can say that the p53 is activated due to phosphorylation. So this activated p53, it is going to lead to certain cellular responses. So there will be three type of cellular responses that can be seen due to the activation of p53. Either it is going to lead to the cell cycle arrest, or it is going to lead to the apoptosis, or it is going to lead to senescence. Okay, so why? Because now the cell has a double-stranded DNA break, which is going to lead to genomic instability. Okay, so we need to activate DNA damage repair pathways. So we need to repair the DNA damage. The very first thing to ensure that the cell doesn't start uncontrollable growth, it doesn't start uncontrollable proliferation, is that you first repair the DNA damage. Or what if the DNA damage is irreparable? If the DNA damage is repair, you just kill the cell. Okay, so if it is irreparable, there's no option to kill the cell, because we don't want the unnecessary defects to accumulate in our body, right? So we kill the cell. So we lead to the programmed cell death. So we choose the apoptotic pathway. So either repair the DNA damage upon sensing that there is some DNA there are some double-stranded breaks, or if it is irreparable, kill the cell. Or you can erase the cell forever in whatever stage it is right now. So this leads to senescence. Okay, so those are the three things that we can do upon sensing that there's some DNA damage due to the certain cellular stress, certain oxidative stress. There is some double-stranded breaks in the DNA. So you have to take those measures as prevention of the normal cell getting converted to a tumor cell. So those three types of cellular responses, they are seen due to the activation of p53. I hope that's clear. So what happens is that under a normal scenario, when the cell is very when the cell is very normal, the genome is quite normal. Okay, under the normal condition, MDM2 will degrade the p53. How? By the ubiquitin annihilation of p53. But when there is some DNA damage, the p53 degradation is stopped. How it is stopped? By the phosphorylation of p53. So this phosphorylated p53 cannot be recognized by MDM2, and this this leads to the three types of cellular responses. Depending on what condition, how much damage is done to the cell, it can either arrest the cell in the cell cycle, so that arresting that, after stopping the cell cycle from going to S phase, it can repair the damage. Or if the damage is irreparable, it is going to lead to apoptosis, or it is going to lead to the senescence. Okay, so these are the three types of cellular responses that can be seen after the activation of p53. Okay, so let's see how those cell cycle arrest and apoptotic pathways are, uh, they're facilitated by the activation of p53. So what happens is that p53 can activate the p52. Okay, so the p53 activates p21, and this p21, it is also known as CKI, cyclin dependent kinase inhibitor, cyclin dependent kinase inhibitor. Okay, CKI. So this inhibits the cyclin dependent kinase, the CDK4/6. and cycline. So the CDK4/6 inhibited, so they cannot form the CDK4/6 cycline D complex, which is very crucial for the cell cycle progression from G1 phase to S phase. Okay, so due to the inhibition of this CDK, so this CDK is inhibited by the P21. Okay, and that is this is how the cell will be arrested in the G1 phase. So once cell is arrested in the G1 phase, now the cell can do the DNA damage repair. Okay, and once the DNA damage is repaired, now the cell will be allowed to enter into the S phase. So the cell cycle arrest by the P21 activation. So let me write it in a different color. P21 mediated cell cycle arrest facilitates facilitates DNA damage repair before cell enters the S phase. So this ensures that before the replication of DNA, the DNA damage, whatever double-stranded breaks were there in the DNA, that will be repaired. So now this will lead to the genomic stability, and this is why we call the p53 as the guardian of genome, because it is guarding the genome and it is not letting it get unstable. So this is why we call it as a guardian of genome, because it is going to arrest the cell cycle in the G1S phase if there is some DNA damage, and it will facilitate the DNA damage repair. How? By the activation of P21. Okay, I hope this is clear to you all. The other cellular response that can be seen due to P53 activation is the apoptosis. So what here in this P21 activation is done in the scenario if the DNA damage is repairable. But if due to certain reasons the DNA damage is irreparable, then it leads to the apoptotic pathway. So what happens is that P53 activated P53, it will lead to the activation of BAX and Puma. Okay, so this BAX and Puma, upon activation of those BAX and Puma, they are going to they are going to release a mitochondrial protein known as cytochrome C. So they will be facilitating cytochrome C release. And this cytochrome C release leads to the caspase activities. And we know that caspases, they are responsible for the apoptosis. Do not fret if you don't know the apoptotic pathway. Just for now, just remember when we will discuss about the hallmark of the cancer cell, that how they escape the apoptotic pathway. In that lecture, we will discuss about the apoptotic pathway in detail, the cytochrome C release, the caspase mediated apoptotic pathway, and the receptor mediated apoptotic pathway. If you don't know, so don't fret about it. For now, just remember that whenever P53 is activated, it is going to activate BAX or Puma, and this BAX and Puma, they do the cytochrome C mediated apoptosis. Okay, for if you don't know the pathway, don't worry about it. We will be discussing those pathway in detail. So this is how through the activation of BAX and Puma, they are going to lead to the apoptosis. Okay. The third, and if there is continuous P53 mutation, there's continuous P53 activation, it is going to lead to the irreversible. So this cell cycle arrest, which is mediated by P21 activation, this is reversible cell cycle arrest. Okay, but if there is continuous activation of P53, the P53 is getting activated again and again, the activation is quite sustained, it is going to lead to the irreversible cell cycle arrest. which is known as senescence, and which will eventually lead to the death of the cell. Okay, so this is how the activation of p53 ensures that the genomic stability will be maintained. Either it gives the cell a chance to repair the damage, or if the damage is irreparable, it just kills the cell. Okay, so this is how the genomic stability is maintained by a wild type p53. So how does it play a role in cancer? So if the p53 is mutated, okay, so the mutant p53 which has lost its tumor suppressor function, this will lead to the cancer. Okay, so the mutant p53 has lost its tumor suppressor function, so it is not going to activate p21. So due to inactivation of p21, the cell wouldn't have a chance to repair the DNA damage, and now the cell will not be able to repair the DNA damage, and without repairing that damage, it is going to enter into the S phase. So now the DNA replication will happen in the S phase, but whatever DNA double-stranded DNA breaks were there, they will not be repaired, and that is why whatever the mutation, whatever damage was done in the DNA, that will be collected in the next generation as well, and that is going to lead to the genomic instability, which will further lead to the cancer. So this will lead to the cancer progression due by by genomic instability, due to the genomic instability. Okay, so mutant p53, it loses its tumor suppressor function, and it cannot activate p21. So it also cannot activate Bax and Puma. So there will be inhibition of apoptosis. So the cells which are damaged, which are defected, which have defects in its DNA, they will not die, and they will accumulate all of the genomic instability, and that will lead to the cancer. So inhibition of apoptosis is seen in the mutant p53, and mutant p53 has a very important and characteristic function, that it shows gain of function of oncogenesis. Okay, so this is very crucial, because this is not generally seen in any tumor suppressor genes. Usually, how does a tumor suppressor gene mutation leads to the cancer? So there is a loss of function mutation. So the tumor suppressor gene loses its function of suppressing tumor. Okay, so there will be a loss of function mutation in that tumor suppressor gene, which is going to lead to the cancer. But this is a very unlikely case in the p53, in the TP53 gene, that it even has the mutant p53. It shows two types of mutation, like it shows two things. The mutant p53, unlike wild type p53, it loses its tumor suppressor function just like any other tumor suppressor gene mutation, but it also shows a gain of function of oncogenesis. Means it facilitates, or it promotes oncogenesis. Okay, so in normally tumor suppressor genes, they undergo loss of function mutation. Okay, so they loses their function of suppressing the tumor. So whatever inhibiting activity that certain tumor suppressor gene is doing, let's for example say retinoblastoma. Okay, in the last class we discussed about retinoblastoma. What does retinoblastoma do? It holds back the E2F transcription factor. Okay, and this E2F transcription factor is the one which is controlling the DNA replication genes. So due to holding back this E2F transcription factor, retinoblastoma is ensuring that the cell will not be able to undergo the DNA replication, right? So this is a tumor suppressor gene. How it is going to how is it suppressing the tumor formation? It is ensuring that the cell will not be able to transcribe the DNA replication genes, and this is how it is suppressing the tumor. So it is ensuring that a cell will not be able to proliferate uncontrollably. And when this retinoblastoma loses its function, it will lead to cancer. So this is a general scenario in any tumor suppressor gene. But in the tumor suppressor gene TP53, which encodes for p53 protein, alongside the loss of function of the tumor suppressing, so alongside the losing the tumor suppressing function, it also gains the function of oncogenesis. So it promotes the oncogenesis. So what it does, it facilitates or it overexpresses certain oncogenes. Okay. How is oncogenesis promoted? By the overexpression of oncogenes such as c-MYC and TERT, TERT. We will discuss about TERT. This is a telomerase subunit. So we will discuss about that, and those the overexpression of TERT and c-MYC. c-MYC is an oncogene, okay? So c-MYC and TERT overexpression, that is going to lead to the oncogenesis. This is a very important point in the p53. So the mutant p53, it is going, so it is it will show the it will be losing its tumor suppressing function, and that is why p21 will not be activated. So it will also lead to inhibition of apoptosis, and that is why the cancer will be seen in the normal cells, and the normal cell will be converted into tumor cell. And again, mutant p53, it will show the gain of function of oncogenesis. So it will promote the oncogenesis by overexpressing certain oncogenes like c-MYC. So due to the overexpression of those oncogenes, obviously there will be a sustained proliferative signal which will lead to the cancer. Okay, I hope everything is clear to you all. Let's summarize it once. Okay, so what we discussed in this lecture, let's summarize it. P53. This is a regulator of cell cycle progression, genomic stability, and apoptosis. Okay, so whenever there is a DNA damage, or let's say there is an oxidative stress. Okay, so this is going to lead to the activation of P53. Okay, so degradation will be stopped, means the p53 is getting activated. So once p53 is activated, it will either activate p21, which will arrest the cell cycle in G1 phase, and due to the G1S arrest, it is going to do the DNA damage repair. And once the DNA damage is repaired, the cell can now enter the S phase and continue proliferation. Okay, and it will gain back its normal function, or it can lead to the activation of Bax gene or Puma, which is going to lead to the mitochondria mediated apoptosis. The cytochrome C is the mitochondrial protein. Okay, so it is going to lead to the mitochondria mediated apoptosis, the programmed cell death. Because now the cell cell cannot, it cannot repair its DNA damage, so it is better to kill that cell. Okay, so either it will allow the cell to repair the damage, it will give it a chance to repair the damage, or it will kill the cell. Or the third cellular response that can be generated by p53 is that it can lead to the permanent cell cycle arrest, which is also known as senescence. Okay, this is permanent cell cycle arrest. Okay, but wild type p53 has those normal, it shows those responses, but unlike that, mutant p53 shows opposite cellular responses. It inhibits apoptosis. Okay, it do not activate p21, and it activates the oncogenes such as c-MYC and TRT activation. So there will be overexpression of those, which is going to lead to the cancer. So I hope this is clear to you all. In next lecture, we will learn about other hallmarks of cancer. Till then, see you. Bye bye.